So let's get started. Hallmarks of cancer. So this workshop is called the Hallmarks of Cancer. And many of you are already experts in the hallmarks of cancer, but maybe others of you are not so much experts in the hallmarks of cancer. And that's what I like to use this lecture for, so to introduce what the hallmarks are. In particular, I mean, so these are 3 papers, right? These were Hanahan and Weinberg. They published 3 papers. One was in 2001, one in 2011, and one in 2022, almost every 10 years. Where they first had 6, and they had 10, and they had 14 hallmarks. And the idea is, what can possibly go wrong? At least for me, that's my understanding. So what can possibly go wrong in tissue or with cells replication so that cancer can develop? And these 14 hallmarks are important points to consider. For me, as a modeler, as a mathematical modeler, this is important to see how biologists think, right? So to experience what do they think is important. And then this should be important for me as a modeler as well, right? Of course. On the other hand, it gives a very nice classification, because then you can refer back and you say, okay, I focus on item 7 and maybe include a little bit of item 8 or so, right? So then you have a nice framework where everything takes place. Okay. But before I talk about the hallmarks, I wanted to talk about cancer modeling in more general, again, from the point of view of a mathematician or computer scientist or statistician, and see what we can do. So where do I start? Let's start here. And there's already a paper from this year, which I really like to recommend. It is by Kulessa.
For the online people, we can actually only see the middle two boards.
Middle 2 boards. Okay, then let's use the middle 2 boards.
Yes, thank you.
There is a way to switch this. This was a button here. So we figure it out later. So let me start here. So this is a wonderful paper in the Journal of Pharmacology, Journal of Pharmacokinetics and Pharmacodynamics. In this year, 2024. And I don't remember the exact title of this paper, but I'm sure you can easily find it. It's about mathematical modeling of cancer. It's essentially the question, what can mathematical modeling of cancer actually provide to us? So what can it help? How can it benefit the treatment of cancer? And this is so important because the authors are all pharmacologists. These are not mathematicians or computer scientists. They all work in pharma industry, so they want to develop treatments. They develop drugs, they develop radiation treatment schedules, they develop chemotherapy agents, etc., etc. And so they ask the question, hey, mathematicians, what can you do for us? How do your results need to look like, or how do your models need to look like, so that we can benefit and make something useful for the patients? And then they have a nice citation about what tumor actually is. I think this is not new for any of you. So a tumor is not just a mass of identical cells, but rather a dynamic population of heterogeneous malignant cells that interact with each other and their microenvironment in ways that influence their growth, survival, and evolution. And there are many, many hallmarks already mentioned in this sentence. So first of all, it's a dynamic population, right? Cancer develops. It is not static, not a static disease, which means our treatments and our models need to be dynamic as well. It's about heterogeneous malignant cells. So it's a big variety of very different cells, of very different phenotypes and very different genotypes as well. It talks about the microenvironment, right? The microenvironment includes immune cells, extracellular matrix, growth factors, blood vessels, stroma, and all that kind of stuff feeds back into the cancer growth. And then it talks, of course, about evolution. So somebody said, I don't know who the citation is, nothing in biology makes sense except in the lens of evolution. So I think this is also true for cancer. So, which means we are in trouble. This is all way too much. So let's talk about cancer modeling. So thinking about this statement a little bit more, or reading this paper a little more, you quickly encounter that we have to work on multiple scales, right? It starts with the genes, mutations play a role. So genes, proteins. So genes are responsible for protein expression, right? And then we come to cells. Or then we have, and then we come to tissue, and then we come to the microenvironment. And then to the patient, to the full body. So there could be systemic things as well.
About the organ.
And the organ, you want to put the organ somewhere, okay. And thank you, Cassia. And I like to invite everybody, please contribute, interrupt, extend, and so on, so that we get a really good picture of all this.
Patient population. All the treatments are defined for the,
Okay.
that's for individual patient at the moment.
Whatever mental factors.
So on which level shall we model this then? I mean, these are like orders of magnitude, right? From genes to populations. I mean, that's impossible, right? So, or maybe not, I don't know. Paul, you can model on any scale, right?
Down level, all that individually would be a bit much.
Yeah, very good. So then they, so these authors, Kuleshta and so on, they kind of identify two ways how we can get a handle on this type of modeling, and they just call it top down and bottom up. So what do they mean by this? So top down, they just mean the data inform the models. So there seems to be a very natural approach, right, in biology or medical sciences. Whatever data you have, I mean, this is what your model should do, right? Your model should not try to describe more than the data really have for you. So if the data is about, let's say, a tumor volume, but it does not measure the immune system, then if you have a model which includes the immune system, doesn't really make a lot of sense. The bottom-up approach, yeah.
Why is that? I'm not sure. I don't get it. I mean,
yeah,
like we had a paper on where we didn't have immune data. We had only volume cancer data. We had some hypotheses about how the immune system is being affected and vice versa affects the cancer.
Yes.
Those are hidden variables in your model, but you can still do systems identification, reverse engineering of variables that are not being observed. That's all the time that mathematics, right?
Yes, very good. Okay. So this would be called the bottom-up approach. So
sorry?
The second approach here, the bottom.
No, but I'm saying if we say there shouldn't be any, the immune system should not be in there if there's no data about the immune system, I completely disagree with that. Because
no, but I think Thomas is saying that that modeling framework would fit in the bottom-up category, right? Is that right, Thomas?
Yes, exactly.
Okay.
So let me just fill in the bottom-up,
Okay.
and then I
Okay.
come back to your question. So bottom-up, if a biological process informs the model.
that's nice questions.
Okay, and I think Eduardo, this. Oh, you broke the chair.
Yeah. No, it's okay. It's okay.
Right, but what you say would fall into this category. So
Okay. Okay. We put it on the side.
So what you said, Eduardo, is this case, right? Where you know the biological process of the immune response is important, and you make hypotheses about how it works, and then you include this in the model as well. So this is what they call the bottom-up approach. But of course, then you have a combination, right? You use the data and you use physiology.
So what's a data-informed model that is not what I'm in this? I mean, I think of data-informed as machine learning, not as.
Oh, I see.
Yeah,
Okay.
So
Yeah.
anyway, it's okay.
Talk about combination. It's a problem of scale separation. But it's not all separate.
Yeah.
And data doesn't need to be in the sense of machine learning. Data could be also understood in the sense of information that fits the model. It doesn't need to be data in the sense that.
No, I understand, but then what would be a top-down?
Yeah.
Down was more of a holistic model.
Yeah.
Which is taking into account maybe as much of biological processes. Bottom up is much more parsimonious, where you're building up building blocks one by one. And they're both top down and bottom up are informed by data, but it's just system versus.
Okay. Yeah.
But that's maybe my understanding. Yeah.
The same as mine. The top one is that then you have the macro perspective and stuff, and then fit the model data. But the bottom of the political data.
Yeah, you might start with some gene model, and you have that as your model, and then you can build on. Okay, now it's a cell growing, and now there's this organ, and
So you take it from the scale.
Yeah,
From the point of view, it's a level macro scale or the micro scale, top to bottom. Yeah.
Maybe that's
Yeah.
a good topic for the general discussion in the group
Yeah.
discussion.
Yeah. Very nice. So I like this discussion a lot. This is fantastic. So I'm reporting what they did in this paper by Kulishta. Okay. So I can always blame them. But still, I think this is useful for us because it allows us to say, okay, I focus on the tissue level. This is what I'm interested in. So I do a model on that level and be very clear about this. Or maybe in transition from cells to tissue or something like this. And as you say, I'm also convinced the scales overlap. Yeah, I mean, definitely. Okay. Oh yeah, that was my last statement. So I want to put this here as well. So we need to strike a balance. Yeah, between complexity data availability and interpretability. And I think this is true no matter how you define bottom up or top down, right? You want to be able to actually interpret what you find. You make some useful conclusions. And remember, these are pharmacologists making this statement. So they're not asking, give us a foot model of every process which could possibly be involved. Give us something which is useful, explains the data, and we can interpret in a good way. All right, so are you ready for the hallmarks? Let's go to the hallmarks of cancer. All right. Okay, so there are 3 papers, as I said. So there are 14 hallmarks. And 3 papers. And so this is the first one is by Hanahan and Weinberg. In Cell 2000. I think earlier I said 2001, so it is in 2000. Now I have my notes. So Hanahan, Weinberg is in Cell 2011. And then we have Hanahan alone in cancer discovery in 2022. And actually, I don't know if the reason is maybe Weinberg passed away. Does any of you know this? Or any of the online people? But anyway, so these are the important papers here. Now, listing these 14 hallmarks will be a little bit boring. So I want to spice it up a little bit, use all your unlimited expertise, and write down the logistic equation, u by dt. Yes, let's call it mu u 1 minus u over k, which is a very standard model for growth processes, also for cancer growth processes. Many of you have used it, I'm sure, as components in your models. And so let's say the healthy tissue does this to a carrying capacity. So what can possibly go wrong to make this an unhealthy tissue? And then I want to ask the question, how do the hallmarks change the equation? So let's think about this as we go along. This is logistic, of course. We could use the Gompertz model, right? You know the Gompertz model with the log of u over k. We could use a van Bertalanffy equation. So there are, of course, other options, but I think logistic is a very popular choice. So it gives us something to play around with. Okay, so first one, sustained proliferative signaling. So this means growth signals, right? The cells get signals to grow, to become bigger, to do mitosis, to grow again, etc. And there are lots of growth promoters known. For example, P10, PIP3, PI3K, mTOR. So I'm definitely not an expert in all these. I found them in the paper and I list them here. But please, all of you, if there's anything, maybe an important growth factor I'm missing. I mean, Eduardo, there are many, right? I'm sure. Yeah.
If you don't have a growth factor.
Yeah, okay.
That's right.
Okay, let's do the logistic thing. So where do we put this in the logistic equation? What if the sustained proliferative signaling is upregulated? Mu, right? Yeah, mu would just be increased, right? So exactly. So here we can put in 1. Yeah. Exactly. So second is the avoiding growth suppression. Yeah, so that's the reverse process, right? There's also signaling going around that tells cells stop growing. You're big enough. That's it. And if this pathway is inhibited in some form, they might continue to grow. Yeah, so examples here would be loss of P53 function or loss of contact inhibition. So where would this fit into our logistic equation model? What?
New term.
The new term as well?
You could push it into K, but I wouldn't.
And you wouldn't. Yeah.
Because K ought to be more carrying capacity of the tissue, not predation.
Yeah.
Unless you mush it in there. I guess the good news with the ODE model is there are only two parameters, and all 14 hallmarks fit in 2 parameters. Bad news is they're all mushed together.
Yeah, very good. Right. So let's put this here. But I don't think you can put all 14 in this equation.
I think he's to put it in K, but
Yeah, yeah, I
in
mean,
K, no,
the fun thing is, it's a bit of both, right? Mu is net birth minus death, but you still don't rise to the same steady level, so you have to force it into K, I think.
Well, if you talk about this contact inhibition, limit,
Growth
right?
suppression, yeah.
So then it would be definitely related to
Yeah,
this
mu
can
both.
capacity.
Yeah, it's supposed to mu, but you still end up with the same carrying capacity, but slower. It's a bit unrealistic.
So I think we can put it there too. Yes.
oh, so you mean making mu a function? Making
Yeah,
mu a
I-
function of mu, basically, right?
It's end up with the same steady population, but it's lower in this form. Which I don't think,
It's not the, right?
yeah, it's supposed, yeah, and it ends up giving a higher capacity and a higher birth rate. Net.
Okay, third one, resisting cell death. So here the example is also P53. So this can signal cell death. I want to call it apoptosis. So cell suicide. But this is essentially the same as a growth inhibition, right? So probably the same, right? In mu and in K as well. Yeah. Okay. How do I continue this now? For enabling replicative immortality.
By the way, Heiflich just died.
Hmm?
Heiflich, the Heiflich, the, you know, the
Hayflick.
telomere, yeah, he just
Hayflick
died
limit?
yesterday. 2 days ago.
Oh,
Yeah,
okay.
it was in the news.
So this they relate to the telomeres. So telomerases can add telomeres. To the cancer cell DNA. And prevent aging. So yeah, so I think as far as I know, not much modeling has been done on the telomeres. So that's another thing as we go through this, I'm going to give you pointers of things I think have not been modeled a lot. I mean, you might have seen people who model this, but I have not seen this anywhere. That people have modeled a telomere extension. So where would this enter our equations?
Mu.
Mu again, right? Yeah, exactly.
So is U supposed to be the volume of the cancer or something about a specific cell? Because telomere extension is at the cell level. So
That is true, yeah.
so where I'm not sure I understand. What
So my U is a cell density.
right. So,
So
why
cell per
would
volume, yeah.
why would it appear in mu? Besides, it's a discrete process, right? It's 50 or whatever, right? But, Yeah, it's not completely clear.
No, but it would translate from the cell level to the population level, right? If you have old cells which are not stopping to reproduce, then they reproduce more, so then the growth rate goes up.
I guess if you start with a heterogeneous, non-synchronized population with different ages. Or something.
Yeah, right.
If they all start at the same age, they would not.
Ah, okay, it's a very good point. Yeah, yeah. Okay, so this one is really, well, we have 14 hallmarks, and then I have 4 more. So it will be 0.20 or something. So we should come back. But these are good points and very nice. Okay. Yeah, right. So here you see already one advantage of this list, because as a mathematician, I would just say, okay, let's change mu. Right? I wouldn't divide it into 4 separate cases. It seems to be a little bit constructed. From my point of view, right? But of course, from a biological point of view, these are different processes and there are different proteins involved. And so the change of mu or the change of k can have different reasons, right? Different biological reasons to do that. So 5 then is indeed different. This is angiogenesis. Okay, who wants to explain angiogenesis? Yeah, Mark Chaplin is probably still sleeping, right? It's probably... Mark, are you there online?
Developing a new blood vessel into the tumor tissue.
Yeah, very good. Thank you. Yeah. Right. So this is the initiation of new blood vessels which grow towards the tumor and then supply the tumor with blood. So sprouting of new blood vessels. Towards the tumor. Okay, and then I want to put a few more details up there. So this is it induced by VEGF? So vascular endothelial growth factor and TSP1 signaling. And then, of course, it has immediate consequences, right? It supplies the tumor with nutrients and oxygen. So supplies tumor with nutrients and oxygen. But then it also supplies a route to get chemotherapy agents in there or other drugs which are transported through the blood. So opens door to chemotherapy or other therapies which are transported in the blood system. All right. And modelers often distinguish between vascular and avascular tumors. So the models are often different. So maybe I'll write this down too. Between vascular and avascular. Okay. Okay, so where do we put this into our logistic equation?
In the carrying capacity, I would guess, right?
In carrying capacity. Oh, very good. Yeah. All right. Maybe I so I can switch them now. So this would be 5. Okay.
Tell me to repeat because they say they don't hear us online, the microphone. they were saying we cannot.
Do they, it's probably the audience they don't hear, right?
Right, apply that.
Yeah.
Yes, if you pass the microphone to the audience, I guess that would make it clearer.
Yes, okay, you can do that.
No one to
So you are the microphone person now.
If you want to speak, I will just pass it. I feel like the carrying capacity is. Sorry.
Yeah, right.
I was just wondering if it would be worth introducing a second ordinary differential equation to model the carrying capacity K, since angiogenesis is a dynamic process. It's not constant.
Right, and it's also on the same time scale as a tumor growth, right?
Right.
Yeah. So it's,
Exactly.
so it would make sense. Okay. So how do I do this? I do this here. Close equation for angiogenesis. So you say it could be an equation for the carrying capacity. Yeah. It could even be explicit equations for the blood vessels, how they go in or something as well. Yeah. Yeah, very good. Awesome. Okay. So what do we have at number 6? Oh, invasion and metastasis. Right, so now we look at the global picture, where they spread, how the tumor spreads locally and also system-wide. And in this context, I'd like to just recall the metastatic cascade. Right, so the idea is there's a primary tumor somewhere here, there's a blood vessel, and so then you get local detachment of cells. They start invading locally towards the blood system. Then they intravasate into the blood system, they're transported about, and then they extravasate somewhere, and they form microtumors, and then they might also form bigger tumors, metastasis, somewhere else. Yeah, so let me write this down to have this complete. So we have local invasion. Then we have intravasation. And then we have extravasation. And we have a micrometastasis. And then finally the colonization. Okay, and each process has its distinct characteristics, and each process kind of needs to be modeled, right, in an appropriate way. Yeah. So you're going to put this all in the moon, Paul?
So you also draw the transport there, right? So between the intra and extravasation, a lot can change. They move individually, they move in groups, they have companions in platelets.
Very good, okay. Then we make this 4, 5, and 6. Yeah, then they talk about circulating cell clusters, right? And they can be surrounded by platelets, which kind of provide support. Yeah.
So for forces to use this, it would be an increase in K because of all the new growth sites available.
Okay.
But we've often modeled this as a network of a bunch of these nucleating new equations for the new sites. And then exchange terms.
Exchange terms between the sites, right? Yeah. And you could even add transport equations along the blood vessels, right? With the fluid velocity or something. Yeah. So I think this does require more equations as well. So for metastasis. How about the local invasion? The item number 1, so just the local invasion here. How can we model this?
By PD.
PDE. Who said PDE?
Yes.
Asian-based models, of course, as well. Yeah. Yeah. No, I would, of course, do a PDE. So just at a diffusion term, right?
Just saying that
Yeah.
at this point, we need a paradigm change, because both the individual invasion by mesenchymal-like cancer cells and the growth of tumor, the epithelial one, they necessitate, from my perspective, both PDs and stochastic individual kind of hybrid.
Oh, very good. Okay. Yeah. So let me add this here. So the local invasion, then here you have mesenchymal cells. And then so you have the EMT, for example, playing a role here. Yeah. Yeah. And this needs additional models. So metastasis, EMT, and so on. Nice. Okay. This and this. Yeah, so these were the first 6 of the first paper from like 24 years ago. How are we time-wise? So, Nikos, you are my chairman, right? You have the time. Good, okay. So then, number 7. So genome instability. Yeah, so cancer is known if they start to mutate, then they seem to mutate even more and even more. And the genome becomes much more unstable. And of course, many mutations will be deadly for the cells, but then there might be every now and then a beneficial mutation for the cancer cells. And so this is called the genome instability. Yeah. So cancer increases mutation rate. And then people distinguish between driver mutations, which are mutations that significantly alter the genome and provide a distinct benefit, a fitness benefit for the cells, and passenger mutations, which do not provide such a benefit to the cells. And Bob Gartenby told me that a full-grown tumor has about 600 to 1,000 different genotypes. So this is really huge. There's not just 1 or 2 mutations going wrong, and then so it's really a lot. So maybe I write this down. Full tumor has 600 to 1,000 genotypes. Okay. So where does this enter our equations? This gets really interesting, right? Yeah.
Cellular reprogramming process.
Yeah. So we need a structured model, right? We need a genotype structured model of some sort. Yeah.
Or phenotype structured model informed by genotype changes.
Okay.
Yeah, do you mean like a series of differential equations with unique parameters for corresponding each genotype? Or a simple equation structured by Eight structural models. Oh, okay, yeah.
Yes, we could have a phenotype variable, you call it phi, which could be a continuous variable. And it stands maybe for the growth potential or something.
This is
Yeah.
partial differential equation, space, time, and this phenotype. Is
Yeah.
that right?
Right. But it could also be simpler. It could just be including stem cells, cancer stem cells and non-stem cells. Then just have two phenotypes. Whatever. Yeah. So there are many different versions. Okay, but yeah.
So at this point, I think the biggest problem that we have as a community is the following. If we describe the epithelial cancer cell, epithelial-like cancer cells by density, this would necessarily imply that we have thousands of millions of possible cells. You cannot use density for a few hundreds of cells.
Yeah.
Then when EMT kicks in, the outcome is of the order of a few hundred or thousands of cell particles. What density that it would. So in effect, I think that you can. I think that you cannot use density for all the different phenotypes in this ladder of mesenchymality, if I may say so. Or at least that's my feeling.
So essentially you say the cell number is small. You really need to work stochastic with stochastic model.
Yes. I think the how's that? The jury is not out yet.
But sorry, why can't you have a compartmental model? You have, some transition, EMT transition, MET transition, and then that has the ones that are in the invasive mode, they might have a higher mu or whatever was the parameter, the reproduction rate, right? So you could do it in a compartmental model, where this second compartment has a very high reproduction rate. For example, I mean, it's not really a reproduction rate, but it's because they transfer to other places, but then those new sites will tend to grow faster. So
yes,
I mean,
but you can do things like that. This is different, and this is what it is being tried. I think that even Paul will speak about that in his talk, and I will do the same later on. It is something that it is being tried, but the jury is not out yet. Currently, these things are published.
okay.
So it means that there's no consensus yet from the community.
Yeah.
How exactly things work. That's my perspective.
Yeah. Okay. I mean, one other aspect that is not mentioned
It's
here.
all about the numbers. That's my point. It's all about
Yeah.
the numbers.
No, I understand. The numbers are
Millions of cells. You cannot describe them individually in kind of a reasonable computational time. Millions upon millions. Whereas you cannot use density on the other end to describe 20 mesenchymal-like.
No, I understand. But if you had a separate compartment for those, that would take care of that.
This is the yes, right. This is what the community is trying.
I was going to say the other aspect that we're missing there is the whole signaling aspect of how the MET transitions, vice versa, how they happen.
Yeah.
MI22, all this micro RNAs and everything, right? So, and people are doing a lot of those models.
That's right. Yeah.
That's missing this picture too.
Okay.
You have the partial derivative over there.
Say it again.
So don't you need a partial derivative over there? Or not?
You mean here?
No, on the left. For the time.
Sure.
It's very going back to genomics and because model mutation is a lot of stochasticity in terms of similar to EMT also in the terms of mutation. Usually there are a few cells that might mutate, and so stochasticity plays an important role. And even they're like,
so maybe
how do you build that into this type of phenotypic structure model?
Yeah, very nice. Good comments. Okay. So then we have number 8. This would be the tumor promoting inflammation. Yeah, so I heard for me, personally, the first time I heard about tumor promoting inflammation was from Laili Shahriari. She's a colleague at University of Amherst in the US. And I think it was 2014, so 10 years ago. I met her at the MBI in Ohio, and we talked about the role of the immune system and cancer. And she told me that there's lots of evidence that the immune system can actually support cancer growth in very different ways. And now, 10 years later, of course, we have many, many examples, right? We have macrophages, M1, M2. We have tumor-associated fibroblasts. We have platelets, which support metastasis formation, et cetera, et cetera. So maybe I write down a few here. Yeah. So inflammation contributes to all previous 6 hallmarks. I mean the first 6. So to the first 6 hallmarks of increased growth, signaling, and avoiding cell death and these kind of things. Yeah, and immune system can make everything work. No, immune system can make everything worse. But I think the immune system is also responsible for all cancers which we don't see. So for all micro cancers which we develop, which are actually not growing into a cancer because the immune system controls them. And this, yeah, I think this is lots. I mean, a person in my age, I'm now 58. I'm sure I have some micro cancers here or there or whatever. And I hope they all will be controlled by the immune system, right? And don't come out too soon or so. But all cancers which we don't see are controlled by the immune system. Okay. Yeah. Now, the modeling of the immune system in itself is huge and big and complicated, as many of you have done and involved with. I say already here that this should impact all the previous 6 hallmarks. So then it impacts the equation here and everywhere, here and here and here. So we have 8 and 8. And then we can have models for the immune system.
Looks that we have a new compartment in the model that's diagnosed and undiagnosed, like basically we have 2 different clusters or compartment that some cancers are not diagnosed and some are diagnosed. Those who are not diagnosed are solved by the immune system or they controlled by the immune system.
Yeah, they might be just too small. They're just lost. Yeah.
Yeah, I was thinking that, like, exactly on that point, that the immune system is kind of fitness pressure that selects for, well, that's maybe not a nice way of putting it, but it kind of selects for these horrible cancers that the immune system can't deal with. Yeah, so it's kind of, as you say, always eradicating these micro cancers, and every once in a while, number 7 springs up a horrible mutation that the immune system can't deal with, and so you kind of prune away all these nice cancer cells that it can deal with, and you're left with this horrible one that will take hold, right?
Right.
It's kind of this positive and negative feedback at the same time, right?
All right.
From that point of view, is also changes to the immune system with age. So many people have been studying of how the immune system, the spleen changes over age, and that can drive then the emergence of new cancer without the need of actual mutations. So there is like heterogeneity and all of the complexity also
For sure,
from that
yeah.
side. So more patient level maybe also on that point.
All right, this is number 9. Energy metabolism. So yeah, so here I definitely need the help of the experts. I want to talk about the Warburg effect and about OXPHOS. And so the normal energy metabolism in mitochondria uses oxidative phosphorylation. And then it produces 30 to 32 ATPs from this process in energy. Whereas the Warburg effect, people are also, I think it's just called fermentation, essentially, produces 2 ATP. And many cancer cells use a second effect, which also works in acidic, very acidic conditions. But so I'm stumbling around a little bit. So let's maybe everybody chip in and help me to get this right. So normal energy metabolism. Is the OXPHOS, oxidative phosphorylation. Yeah, so everybody or people say it provides 30 to 32 ATP, but I kind of couldn't get it. So what produces 30 to 32 ATP? So what is the input? Is it sugar? Right, glucose, right? Yeah, okay. 30 to 32 ATP. Okay. And then the other one, the unnormal, I don't know, it's probably also normal. Anyways, so it's called the Warburg effect. This is aerobic fermentation. And so this produces 2 ATP. So from the energy balance, it's much less efficient. But normal cells usually don't do this, but cancer cells can do this, and this gives them a selective advantage in hostile environments. So it can lead to resistance for drugs. It can also.
I just wanted to add that the second thing, the fermentation is much faster. So you get an advantage.
Okay.
So the oxidative processes, they just take longer. And this 2 ATP you can produce, I mean, you produce just 2 of the ATPs, but you can make it much faster. So you create a conveyor a little bit. And that's the advantage that you can get.
Nice.
And
Okay.
yeah.
Very good. So much faster.
Yeah.
But in the aerobic respiration, so when 2 ATP is produced, so in byproduct lactate and H plus ions are also produced, and these are maybe helpful to local inhibition and such things.
Okay. So it produces a local acidic environment, which then is a disadvantage for healthy cells and another advantage for the cancer cells.
Yeah, I think the previous point about the speed with which the 2 ATP are produced is a crucial factor, because I think we looked at this before, trying to look at the energy expenditure over a bigger field than just that local equation. And I think it seemed that this Warburg effect, or shifting to the glycolytic metabolism, is actually advantageous, the energy-wise, you know? Although on the surface, it looks like, oh, you're getting 32 ATP for OXPHOS and only 2 ATP. Why does it shift? But actually, when you try to model the energy expenditures on a slightly bigger framework, I think we found this glycolytic metabolism is advantageous for cancer cells.
Yeah, thank you, Sid. This is really interesting. Yeah. Okay. So I think the modeling on this regard is not too much. I mean, Siv just mentioned he has done some modeling there, but correct me, Siv, but I think there is not much, right?
Yeah, you're absolutely right. There's really not much. Yeah.
Okay. So back to our equations. Energy metabolism, where will we put this? I mean, you need energy for growth, right? So let's certainly put this here. Otherwise, it's a phenotype. It's a phenotypic switch, right? So then there would be, phenotype structure things.
Neglecting that differential coefficient, like in terms of the speed of mutation, or something like the energy might come there.
Okay.
I'm just guessing it. Oh, sorry.
Just guessing that maybe the diffusion coefficient might be affected by some of these hallmarks.
I certainly want to have the metastasis and local invasion there, and the diffusion coefficient and the energy metabolism as well. Okay.
Right.
Yes, Julia.
There was some work by on how the production of the local acidic environment actually aids invasion as well.
Yes.
So from that point of view.
Okay. Yeah, so I keep this here as
Yeah, knowing that.
Yeah.
Also.
Then other slides.
I mean, there's been quite a lot of work on glycolysis and EMT and people tending to model it as an emergence of a second population that can survive where the first population can't. So I wouldn't say that it has not been modeled very much. And certainly the bioinformatics folks have done tons of, transcriptomic analyses on these populations. So I wouldn't put this in the rare modeling category.
Yeah,
if K is spatial, then effectively it says for those cells, the carrying capacity is increased because they're not dying there. Now, it opens up new niches and gives a competitive edge to anyone who has not, against any cells that have not adapted to that acidic environment. So there are a few dimensions in that equation, and it's definitely been looked at.
Okay, very good.
Yes, I think I was really talking about an explanation of the Wber effect. Why cancer cells switch to that glycolytic metabolism. I don't think I've seen really anything much on trying to answer that through mathematical modeling.
The sound is wonderful. Yeah. No, that's true.
It may not be. It may not be that they are specifically trying to only turn this one thing on, but it comes as part of the EMT program. So they're more trying to unlock something else or just increase variability, turns on a bunch of programs. This one just happens to work in a few places. Maybe it's not advantageous all the time, or it's not selected for individually, but more as a downstream effect of other things that were turned on.
Right. Okay.
And of course, could be the microenvironment too, right? Which turns it on. Yeah. Okay. Good. Move on to the number 10. 10 I squeeze in down here, which is avoiding immune destruction. So evading immune destruction. This sounds a little bit similar to the immune response which we had earlier, but I think this relates to a little bit different process, this TP1, TPL1 process, where we have active signaling which deactivates the response of the immune system. So PD1, PDL1, immune inhibition. And this is, as far as I know, currently a very hot topic. Many people, many labs, and also many modeling. For example, one of my students works on. So, okay. So then 10 would be, where do we have the immune system? What was the immune system? 6. 8. 8 was the immune response. So this is then here. 10. And here. Okay, so 11, phenotypic plasticity. I think this is something which we lumped in with the genetic plasticity already, right? So the differences in cell behavior could be phenotypic or genetic or epigenetic and all this. And I think we discussed these things here already in this item. So then this would be 11. Yeah. And then 12 is the epigenetics. Programming. Right, so epigenetics relates to cells which have the same genes, but they still behave differently, which could be related to the microenvironment, or it could be related to the phase in the cell cycle where they are, or their age, or whatever. So, so then this would go in as well.
Something. So, yeah.
I got it. Yeah.
So, why do they divide 11 and 12? No, I should probably, but like, what's the key difference between the two?
Oh, I see here in phenotypic plasticity, they also include de-differentiation. So that you have more differentiated cells which de-differentiate to become stem cells or pluripotent cells, and then differentiate some other way.
Shouldn't that be then better described as the phenotypic elasticity or phenotypic elastoplastic behavior?
I think it's just generally called phenotypic plasticity. I mean, that's in the literature anyway, but.
Yeah, I think you're right.
But sorry, I had the same question that was asked. Why would not that be included in 12? I mean, it's an epigenetic process. It's not genetic changes. Why would it have its own category?
So you have to ask the authors. So yeah, I don't know.
Epigenetic is still heritable, whereas phenotypic is moment by moment. Daughter cells still inherit some of their epigenetic characteristics.
Proteins that are overexpressed and things like that. So I'm not so sure.
Oh, fair. I mean, mathematically, it's just what time scale do you believe these things happen at, I suppose, too, to an extent.
Right. So it's a question of what epigenetics mean. Does it just mean, methylation changes and things like that? Or does it also mean, being in a different domain of attraction of a different attractor, which is a dynamic thing, right? I mean, this really goes back to what is the definition of epigenetics.
And a lot of this is so mushily defined.
Yeah, exactly. Yeah. Okay.
Yeah. Okay.
Sorry.
No. I'm
Okay. Next one, 13, is the microbiome. Of course, the body, right, has many different microbiomes. We have a microbiome in the gut. We have microbes in our skin, in the lungs, and then everywhere in the sexual organs. And I mean, they must have an impact on cancer growth, right? I think this is a very natural assumption, but not many people have looked at this at all. So, yeah, maybe I give you the examples here. So gut, lung, skin. How do they call this? Urogenitals. Okay, so in this context, I can tell you my favorite theory for travelers. So when people travel into different countries, let's say India or China or so, they get upset stomachs, and many people say they have food poisoning. But I don't think it's food poisoning. I think it's a microbiome. I think if you just, let's say, go to India where you have not been in a long time, just the food is different, the enzymes are different, the microbiome is different.
Yeah.
And you need to rebuild your gut biome before you can fully adapt to the food in a different country.
Yeah, Thomas, I think you're right. And actually, there has been, and now recently, some modeling, and we've actually looked at the impact of probiotics and prebiotics on the immune response to cancer cells. So yeah, I think that's an important factor that hasn't really entered mainstream too much as yet.
Very good. Yeah, so that's a very interesting topic. So then, I think this would deserve a new equation. So models for microbiome. That would be number 30. And then SIFs you will find interesting. I also have a solution how you can then deal with this. So if you go to another country, you just should kiss many people. And then the microbiome will adapt.
Okay, I should have talked to you before I spent 3 months in India earlier this year.
Okay, and so then the last one, 14. Senescent cells. So senescent cells are essentially sleeper cells. These are cancer cells which fall to sleep, maybe through external stress or treatment. And then when the treatment subsides, then they wake up and they, hey, I'm still here. And then they can repopulate the tumor again. So yeah, let's call them sleeper cells. I don't know if you've seen the Bourne movies, the Bourne Ultimatum and the Bourne whatever. And he always encounters these sleeper agents, which are just woken up to try to kill him. So that's
Yeah,
it. Yeah.
Excellent series.
So they can avoid treatment, so treatment resistance. Yeah, and for example, tumor-associated fibroblasts are known to initiate new cancers. From a senescent state. I know, Julia, you work on senescence, right? And
Well, I haven't before, but that's what I want to do here. So I guess.
Oh, that's what you want to do here. Okay, so then the 14, we arrange this, so 14 is visible. Senescence, so sleeper cells. Yeah, we need another equation then. Well, it could also be seen as a phenotype, right? So then we could also put this here on the 14. All right. Okay, so these are the 14 hallmarks. So I think I have a few more minutes, at least according to the program. Because I have 4 curiosities, which I also like to mention. But before I do this, any additional comments on these hallmarks? I mean, this is a lot, and this is a preparation for the meeting, right? I mean, we will hear talks of this and that and everything during the entire week.
Hi. Hello.
Just remind us the list.
Show them all of them.
Covered. Yeah. It got covered. Just go quickly over. Look, just asking.
Okay, there was a comment online too.
Yes, hi. Nice talk. I want to know the oxygen heterogeneity is categorized in which hallmark? Oxygen heterogeneity. Yes. Oxygen is crucial element, right, to growth. And also the metabolic switch from one kind of metabolized to second kind metabolized, like the glycolytic metabolism is very much rely on oxygen concentration. So
yeah, I think it would probably impact a number of the hallmarks, right? The oxygen heterogeneity.
Probably. Yeah. But I guess it's the general idea that the microenvironment can affect all of the hallmarks that we have seen, more than being an hallmark itself. It can, well, it's just the definition, but I guess people have mentioned like how oxygen and other factor in the environment then shape all of these hallmarks.
Yeah, and as we talked about the energy metabolism, that there are two types. You're right, a switch is possible, right? So the phenotype can change and they can use a different energy metabolism if they have to. Yeah, that's an important point. Very good. Yeah, there's a nice graphic for the hallmarks, which maybe I show at the beginning of the next talk, and I can upload it and just show it there. So then the four little curiosity items. So one is the oh, right, yeah. So I talked to Bob Gartenby as well. Everybody should. He's really great. So he told me that the hematopoietic stem cells, these are the stem cells which recreate our blood, are essentially cancer cells. They have like essentially all of these hallmarks. So the hematopoietic system, they create 100 billion cells per day. So they're replicative, out of control, they're immortal. They have all kind of growth signaling and whatever going on, because they reproduce so many red blood cells, white blood cells, immune cells, everything, in every single day. Right, so then it means these hallmarks are actually sometimes even useful in certain contexts. So that's interesting.
Yes, Thomas, that's I think what you mentioned about the hematopoietic system, where the cancer stem cells sort of mirror what happens in normal development, is the sort of model that everybody's trying to grab for example, brain tumors or, bone cancers. So I don't know whether it translates as well, but, that's, I think, shaped the thinking of many of the experimentalists and things that in the context of many other cancers.
Yeah, Silva, this leads me to my second item, which is the atavistic theory of cancer. So the atavistic theory, you want to say something? Yeah.
Sorry.
Okay.
Oh, sorry. This still
the hematopoietic cells. I was just wondering if there are a literature of mathematical models developed in that space that we could draw upon or influence our models of cancer. Are there models of hematopoiesis?
Oh, absolutely, there are models. So, for example, Anna Marciniak-Chokra in Heidelberg, she has her career is basically on modeling of that. So,
and is there significant overlap then between those models and the kind of terms that
they then model leukemia, right?
Oh, sorry,
yeah, leukemia cancers. So that
of course, yeah, that makes sense. Thank you. Sorry.
So then the atavistic theory of cancer says it has to do with cancer evolution, but it says essentially that cancer evolves backwards. So that cancer uses properties or traits which it had earlier in the development of the tissue. So for example, brain cancers, when they spread, they use the same machinery as an embryo used to build the healthy brain. And this is also known for other cancers as well. And actually, I know that people work on these evolutionary therapies to prevent resistance. But I've never seen this in the context or in the point of view of the atavistic theory, so that evolution is actually backwards in the cancer and not forwards. I don't even know if this makes a difference from the modeling or not, but I find this interesting.
Yeah, no, Thomas, I find it quite fascinating. I've forgotten. I think it was probably a cosmologist, forgotten his name, Paul Davis, I think it was, who was talking about sort of cancer cells really being like a way of running in safe mode, like a, you know, if you're thinking about a computer operating system when faced with an external threat. So they you know, introduce this quite controversial new theory, you know, exactly like you said, based on its evolutionary roots.
Yeah.
So I think it's a quite fascinating theory.
Yeah, thank you, Sid. Yeah. Yeah, Sid, we really need you here. Why are you not here?
I got tied up with I'll be there on Wednesday, but I just couldn't make it. Yeah.
So then the third item is chronotherapies. So that actually therapies for cancer or cancer growth depends on a circadian rhythm. And a treatment in the morning is different than a treatment in the afternoon or treatment in the evening. And this is not included in any of these here. But of course, there are people who worked on this, like Claire Rambeau or Jae-Kyung Kim in Korea. So circadian rhythms. Chronotherapies. And then one more thing I wanted to mention is metronomic therapies. So metronomic therapy is, the idea is for chemotherapy, for example, you give a low dose, which just controls the growth, but does not eradicate the cancer. And through this idea, you prevent resistance or the selection for resistant cell lines. Now it's known as evolutionary therapy, but I think the idea of metronomic therapy is actually older. And again, I'm not sure if the evolutionary therapies and the metronomic therapies have been brought together in a modern plan.
A different category towards some people called adaptive therapies, where you keep track of the resistance of populations.
Yeah, that's exactly my question.
Well, but that's different. You know, some people go metronomic. There's just a question of semantics. In a different group of people do different things. In metronomic, you don't necessarily.
You don't follow the resistance track, yeah.
Right, maybe an open loop is supposed to be closed loop.
Yeah.
Control rates.
Okay.
Let's include that.
So adaptive therapy. I think actually there's some modeling which could be done as well. Compare the metronomic and the adaptive things. Yeah. Yeah. All right, so that's all I wanted to talk about. Thank you for all your feedback. So it's really wonderful. Thank you.